Okay, we will continue this lecture. So, just now we discussed about the structure and function of compound eyes, that is ommatidia. Now, next important one is based upon the type of images formed by the ommatidia. The compound eyes are divided into two types. One is known as the apposition eyes, second one is superposition eyes. Now, these are the differences between the apposition eyes and superposition eyes. So, if you look at these four differences, you will know what are apposition eyes and what are superposition eyes. So, these differences are very important for your examination. Now, these apposition type of eyes are mostly they are present in diurnal insects. Diurnal means the insects which are active during daytime. Now, these are present in nocturnal and crepuscular insects. Nocturnal means they are active during night time. Crepuscular means those which are active during dusk time. The second difference is the image formed in this type of eyes is because of the light which is received by the ommatidia, and the image is formed by the same ommatidia. Okay, but whereas in superposition eyes, the image formed is because of the light received from different ommatidia. So that is a major difference between apposition and superposition. Now here the pigment cells which are surrounding the ommatidia, so these pigment cells do not allow the light to pass from one ommatidia to another ommatidia. So this is the reason why the image is formed only by the same ommatidia. Now here, because the pigment cells are absent, so the light received from different ommatidia are responsible for the formation of the image. Okay, so this is second important difference. And here the image formed is very distinct. That means it is very clear. The image formed is very distinct and clear. Now here the image formed is not clear, and only the insects can see the general feature of the object. It is not the real image. Okay. Examples are butterflies, and here the example is moths. Okay. Yeah, this is from this diagram you can see the difference between the apposition eyes and superposition eyes. Now here you see the image formed is this is the image. Okay, the image formed is because of the same ommatidia. Now here you see the image formed is because of the light received from different ommatidia. So these are the this is the major difference between the apposition eyes and superposition eyes. Now next important visual organ is simple eyes, or these are also known as ocelli. There are two different types of simple eyes or the ocelli. First one is dorsal ocelli, second one is lateral ocelli. So these are also known as the stemmata. Okay. Now these dorsal ocelli are seen in case of nymphs and adults of hemimetabolous insects. So this is very important. Okay, and also in the adults of holometabolous insects. And generally these dorsal ocelli are generally 3 in number, but in some it may maybe 4 also. Okay. Now the dorsal ocelli which are present in the cockroach, they are represented by special structures known as the fenestrae. So this is very important for your examination. So fenestrae type of ocelli, dorsal ocelli are present only in case of cockroaches. Now coming to the lateral ocelli, these are also known as the stemmata. So these lateral ocelli are very special in that these visual organs are present only in case of endopterygotal larvae, for example butterfly larva. Okay, and generally these ranges from 1 to 6 in number. Now here you can see from this image, you can see, yeah, these are the dorsal ocelli. These are 3 in number. The middle one is known as the median ocelli, and these two ocelli are known as the lateral ocelli, and these dorsal ocelli are generally located in the frons region of the head, and these 3 ocelli are located in between the two compound eyes. Now these are the compound eyes. So in between the two compound eyes, you can see three dorsal ocelli. One is median ocelli and two are lateral ocelli. Now this is the structure of the simple eye. So it is just like the ommatidia, where you can see the corneal cells, then carnesian cells, then you can see the crystalline body, the rhabdom, and the pigment cells, and central retinal cells. Okay. Now next part of this lecture is auditory organs. So what are auditory organs? So these are the organs which are present in at different locations of the insect, and these are the organs which perceive the sound waves, and also from the aquatic water currents. So they perceive the sound waves as well as the aquatic water currents. Okay. There are four types of auditory organs present in case of insects. First one is auditory hairs. Second one is very important auditory organ, tymphanal organs or Muller's organs. Okay. Third one is Johnston's organ, and fourth one is pilifer. Okay. Now one by one we will see. Coming to the auditory hairs, so these are present in case of the larvae of Lepidoptera, which you will see in the images later. Second one is tymphanal organs, or they are also known as Muller's organ, and these tymphanal organs are very special type of auditory organs which are present in case of orthopteran insects like the short-horned grasshoppers as well as in the long-horned grasshoppers. Now as far as long-horned grasshoppers are concerned, so these tympanal organs are located on the foretibia, whereas in case of short-horned grasshoppers it is located on the first abdominal segment. These are very important. The location of tympanal organs in case of orthopteran insects is very important. So in case of short-horned grasshoppers, it is located on the first abdominal segment, and in long-horned grasshoppers, it is located on the foretibia. So these sites or the location of these organs you will see in the images later. The third organ is Johnston's organ, which is also very important organ. This organ is generally it is located on the pedicel segment of the antennae. Okay, these organs are present in almost all the insects except Collembola. Okay, and pilifer, this is also very specialized auditory organ present in case of certain few insects like the Spingidae, which includes the hawk moths. So these pilifer organs they respond to the ultrasonic sounds. Okay. Now here you can see this is a tymphanal organ which is present in case of long-horned grasshoppers. Okay, and the foretibia you can see a ear-like structure you can see. Okay, whereas in case of short-horned grasshoppers, so you can see this tymphanal organ on the first abdominal segment. Here you can see a ear-like structure. Okay, yeah, these are the pilifer organs which are present in the spingid or hawk moths, so which will respond to the ultrasonic sounds. And here also in some other insects here also you can see the auditory hairs. Okay, yeah, with this we have completed the sense organs, and finally there is a quiz for you, so wherein I have included 8 fill in the blanks questions and 2 questions. Okay, so please go through the PowerPoint as well as from the notes, and try to get the answers for this, and also prepare for these questions also. Okay, that is write a brief note on structure and function of compound eyes with the diagrams. So as I said in the last class, so all the diagrams, all the figures which are given in the previous classes are very important. Okay, so I am going to give a small assignment also on this aspect that I will inform you later. Okay, so this is a question which can be expected in the final examination. And second one is of course very important one is differentiate between the opposition eyes and superposition eyes. Okay, so with this we have completed all the lectures.